Good afternoon, and thanks for coming. This is the micro and nanoscale technology session. I'm Hong Lu. I am associate professor in the School of Chemical and Biomolecular Engineering, so I'm sort of chairing this session. We have 4 great talks coming up, and I'm just going to lead off with a little bit of the introduction to what's going on at Georgia Tech and how I see how the micro and the nano world fit into the bioindustry. Am I speaking loud enough for people in the back? Okay, good. All right, so I have a couple of intro slides, and then I'm going to talk a little bit about what my lab does, because I feel a little bit awkward talking about other people's work. I don't do as good a job. So hopefully we'll hear the speakers talking about their work, but I just wanted to give people a little bit of an impression of where I see this field is going. Well, actually, it's a very large field, the micro and the nano technology world. So the grand challenges that I see for biology and for biomedicine are the following. Cells are small, molecules are even smaller. We normally think about biologists in terms of handling flasks, beakers, and whatnot, and putting biological entities in them. So there's a notion in this field that because the entities that you're interested in are so tiny, you probably should develop the right tools at the right length scale for manipulating these things. So in the last couple of decades, there have been a lot of research that is done on creating these micro tools, and in some cases molecular tools, and some people call it nano tools because it's smaller than the micron, in interrogating the cellular and the molecular systems. So yeah, my notion is that the macro tools is probably the wrong thing to use to interrogate the biological system. So the goals in engineering micro and nanotechnology is that, well, there are 2 sets of goals. The first sets of goals are usually involving in getting information about the biological system. And then there's another set of goals is actually sending information to biological system or directing their activities. So in terms of getting information from the biological system, we actually want to know disease mechanisms, how cellular activities are how cells actually behave in terms of in the cells that are normal and the cells that are doing different things or diseased cells in the disease systems. So we want to image molecular and cellular events. So there are a lot of tools that are actually developed to allow you to interrogate what's happening inside the cells. There are also high throughput screening technologies that actually allow you to do things at the proteome scale, for genomics, getting information about systems, behavior, networks of things that are happening inside biological systems. We also want to get information about patients on the patient level, so diagnosis would be one thing that we'd be interested in. And related to that, sensors at all levels, from organisms all the way down to molecular sensors. So in this particular session, you're actually going to hear a lot about how diagnosis on systems level for diseases. Weber-Lamont will talk about how you diagnose diseases in the blood. Sensors and imaging and interrogation techniques. So Todd Solchek's lab and my lab also work on microfluidic systems to interrogate cells and all the way up to small organisms. We're also going to hear a talk from Bao's lab on molecular sensors, or using molecules to interrogate other molecules inside cells. So to me, getting information from biological systems is actually a highly complex problem, and it really involves multiple scales, and it's not something that one technology can really conquer. So I think it really takes the integration of technologies at different levels to solve these problems. So the second thing that I want to mention a little bit, because the information is not just one way, that we're not just getting information from biological system, is that in a lot of corrective situations where you want to cure a disease or you want to direct biologics to happen. So for example, in the morning, this morning I actually attended the drug discovery sessions where people actually use biological systems to make drugs, for example. So in those situations, you want to direct the biological system to behave the way that you want. So again, there you would want to use molecular tools and in a lot of situations, actuators. So that sort of is in parallel to the sensors domain. And at Georgia Tech, there are a lot of people who are actually putting micro devices inside animals or potentially human. So those would be implantable devices. So these are the areas that I think could address the challenges that we're talking about here, how to interrogate biological systems and how to get them to do the things that you want them to do. So opportunities, I mentioned these already, but I just wanted to summarize it a little bit. So there are a lot of opportunities for instrumentation development, and I think I and a lot of the speakers in this session are in the business of instrumentation development. So we're coming up with engineering methods, technologies to get information that you cannot get otherwise. So the advantages of doing microsystems is not just more experiments, faster experiments, although those are things that everybody would want. I think the value, the greatest value of doing microtechnologies is essentially getting information that you cannot easily get with macro scale experiments because you're interrogating the system at the right length scale. So tools to put stuff in and put stuff through. So I think about these things as manipulations to smaller systems like cells or even small organisms. So we develop some of these techniques to shovel things through, to interrogate them. And tools that actually, tools to put stuff, to put into stuff. So this is what I refer to as the implantable systems. So you have microelectrodes arrays that you can stick it into a brain, or other implantable drug delivery systems and things like that, or monitor sensors that you can actually put it into human or animal. So that's what I would call the instrumentation development. And there's plenty of research that's going on at Georgia Tech in instrumentation development. So a lot of electrical engineers and mechanical engineers, I didn't list their names here, but I'm happy to, well, I'll probably mention some throughout this talk, but the MEMS people, there's a session tomorrow that talks about MEMS technology and microelectronics technology. So there are a lot of those things that it can be used as implantable systems, cochlear implants, ultrasound probes, et cetera, microneedles. So the other category of research that's in comparison to the instrumentation development is point of care diagnosis. So we're going to hear Wilbur talk about this later on. So this is a class of technology where I think is cheap and it's also easy to manufacture, and you can actually take this to, easy to use as well. So you can take this to either doctor's office or a bedside, or actually deploy it in the field. And the instrumentation requirement there is going to be vastly different. The type of technologies that you want to develop is also very different from what you do at the high-end scale, where you have all the laboratory tools that are available that you can use in your own laboratory. And the other 2 points that I want to make in terms of opportunities, that I really view these problems as an opportunity to integrate multiple scales. Like I said earlier, molecular tools are often integrated with cellular tools or with instrumentation tools. They don't really operate in vacuum on their own. So I think there are a lot of collaborative opportunities from industry and also among the academic labs to integrate all these things together to solve a biological question. So molecular probes, there are several labs on campus that actually make these molecular probes and doing molecular imaging. So Phil Santangelo's lab, Christine Pang's lab actually do these imaging at the subcellular or molecular level. Genetically engineered tags, and earlier in the morning there are a lot of labs that actually make these genetically engineered system to follow metabolic pathways or follow molecules and how they traffic through the biological systems, or potentially drug delivery, and you want to know where the drugs are. Biomaterials field would fit into this category. As well as analytical tools. So mass specs, so there are several mass specs lab that are on campus, and I see this as an opportunity to integrate it with perhaps MEMS technology or microfluidics technology. So the analytical tools would be downstream from the cellular processing units. There are plenty of opportunities for collaboration on campus. And then the last point that I want to make here is that I really feel that this is not just an engineering problem or a bioengineering problem even. I think it does involve a lot of inputs from the chemists making molecular probes, potentially, doing genetic engineering, computer scientists to automate things, to do things more quantitatively with these large data sets that we have to manage, and statistics. So it really takes all these people knowing how other fields work and where the opportunities are and put the expertise together. So the last slides that I have, I just kind of want to mention that in the micro and the nano world, I think these are where we actually could make some differences. The areas in biology include neurodevelopmental diseases, so my lab does a bit of that, neurodegenerative diseases, immunology. There are several labs on campus who work on immunology. Cheng Zhu's lab, Melissa Kemp's lab, Julia Balvinsi's lab, who address these diseases, autoimmune diseases and aging of the immune system as an example. So stem cells and regenerative medicine. I think this is a field where you have a lot of cell biology to do. You have a lot of interesting problems where you want to look at cohorts of cells. You have opportunities to do genetic engineering and also drug discovery using these platforms. Biomaterials world, so how cells interact with microenvironment. And I think microfluidics and MEMS technologies could be a really great place to actually integrate materials and cells. Cancer biology. Okay, so well, I won't go through the details here. I'm just going to now, I don't have a whole lot of time, so I will show a little bit of the research that's going on in my lab as an illustration of these points, where the research is really multi-scale, and we really want to create an environment where multiple disciplines come in and solve these biological problems. So my lab is not so much on the applied side, meaning I don't make devices and put it into mouse or human systems. We're really on the fundamental biology side, where we try to create instrumentation that address problems that are hard to do with regular conventional technologies that are in everyday lab. Because Todd's lab and Wilbur's lab also work on microfluidics, I'll just give this one slide of intro. So essentially, this whole field of microfluidics really come from the analogy in the microelectronics industry. So there, people 40, 50 years ago have decided that by integrating transistors, making them onto these chips, you can reduce the cost as well as computing speed and increase the integration of functionality onto a chip. So today, one of your iPhone chips or whatever it is, smartphone chips, would be a lot more powerful than the earlier supercomputers in the good old days. So what we were trying to do in the microfluidics world is also to follow suit, essentially thinking about shrinking down the laboratory manipulations of cells, molecules, and maybe organisms down to these smaller chips and put a lot of functions onto one chip. And that way, the volume of these very costly reagents will be reduced. You can integrate functions, you can do things faster, and there's actually, in a lot of situations, interesting physics that can be taken advantage of to do things better. Okay, so that we will illustrate it with a few examples, and you'll hear throughout this session. So this is just a simple illustration of a sequencing center back then would require, a ginormous facility, and now you can actually do digital PCR on the chip that's about 1 inch by 1 inch. Okay, I'm just going to introduce one organism that we work on, because this is not something that a lot of people are terribly familiar with. C. elegans. So this is a little worm, a nematode, that has a very simple nervous system. So we're interested in the neurons in this system, how the neurons actually come about, how do they form synapses, and how do they function. This is a nice system because it's very tractable. This worm is really cheap to grow, growing on the bench, and it has exactly 302 neurons out of 959 somatic cells. But it's very informative. So this worm actually carries a lot of the genes that have, a lot of the worm genes are ortholog to human disease genes. So somebody estimated that are about 60% of the human diseases that have genetic origin would have genetic orthologs in worms. So we can use the system to do drug discovery, to do genetics, genomics, actually, discovering these fundamental mechanisms that are governing the biological system all the way from the lowly worms to flies to fish to chick to mouse to human. Because biology usually doesn't reinvent itself. Evolution sort of set up things that work pretty well at the lowly worm level, and then these mechanisms get conserved. Okay, well, I have no idea why this slide jumped over here, but okay. So one thing that we're interested in is to look at the genetics and genomics and doing drug screens with the worm system. This is a review paper that I lifted, and I got 2 pictures. This is actually a slide I borrowed from a student. And the idea is that there are these screens that are from hell and screens from heaven. So screens from hell are the screens that actually well, screens from heaven are the screens that are really easy to do. You're looking at very obvious phenotypes, whether the worms are dead or drug resistant, or whether they grow, or whether they actually even crawl normally or not. The screens from hell are things that are looking at very fine phenotypes, things that are of interest. So nowadays, the low-hanging fruits are gone. The screen from heaven are all not quite there anymore because people have been working on this system for about 40-something years. The interesting things are what we call the screen from hell. Very difficult to do. I'll give you one example. We're interested in the synapse formation. So we want to actually look at these structures that are smaller than the micron in the worm's nervous system that we tag with a GFP. So the manual methods that almost all labs in the world who work on synaptogenesis in worms would do is to mount the worms on these slides and look at them. So I think some pharmaceutical companies, if they use worms as a platform for drug discovery or studying fundamental biology, would also do this. There is a system commercially available for high throughput imaging, but this is a system, essentially, it's a modified fluorescence activated cell sorter. The only issue with the COPAS system is that it's a very low resolution system. It's a one-dimensional system. It doesn't give you resolution down to be able to see synapses, for instance. Throughput is very high, but you just don't get the information that you want. So what we came up with are these microfluidic chips, essentially to handle the worms, to allow you to look inside and then interrogate the worms using the optical techniques that are available for the manual methods, which is whatever you can see, diffraction limited optics. So these this is an example of a chip, and it tells you how big it is. The way to think about it is that these are essentially smart cover slips that you mount onto a microscope, and then they manipulate the worms as the worm comes through. So I'll show you a movie. This is how a very early version of this device works. So we route the worms through. They park over there in that region for a couple of seconds, and then we can grab images and analyze them and decide what to do with them. So this is a real-time movie, and usually you can get a throughput of 1 worm per every few seconds or so. And just to give you a sense of scale, this cross dimension is about 100 microns. So this channel right here is about 100 microns. So if you put a piece of hair under the microscope, that's how big it is. So my student May will talk a little bit more about this later. So using this particular system, we actually integrated a lot of quantitative imaging techniques, and as well as computer science techniques such as artificial intelligence, which is essentially a way to mine the information out of these pictures that we take. Because in a lot of situations, the humans are actually pretty bad of getting quantitative information. Things that we're interested in involve very subtle changes in the things that we're looking at. Synapses. How big are they? How much smaller that they are compared to the other worms? How submicron, in terms of submicron locations, do they shift along the axon of the process? Or how many slightly bigger ones of these synaptic puncta that we can actually observe in the mutant worm versus a wild-type worm? You could imagine that you could do drug screens or RNA screens. These are genome-wide screens with a system like this. So without telling you too much about it, we just published this thing. This is essentially integrating all these technology to make a system that allow you to, so this picture is essentially showing a bionic eye of the worm. The idea is that you will use electronics, a computer, to think about the information that you're getting while imaging a worm through our microfluidic system. And the ability to make decisions at that point is going to be a lot better than the human calling the shot, calling whether the worm is a score or the drug is a score, based on subjective information. So again, this is a cartoon showing the idea. A worm comes in, and then you decide whether the worm is of interest or not, and you tell the worm to go one way or another, like in the device that I showed you earlier. So in the end, we really would want to transform the idea of doing experiments with a biologist to making decisions by a computer. So does anybody know what that symbol on the right is? That's Watson. So that's the IBM supercomputer that actually is now able to process loads of information and answer questions in a humanistic way. So essentially, that's using artificial intelligence to make decisions. And what we're doing here is transforming the way that biologists do experiments. By the way, this is a picture from a couple centuries ago, what a biology lab looks like. And if you walk into a modern biology lab, the only updates is probably the microscope. Everything else looks fairly similar. So what we're really trying to think about is to use technologies, use the microtechnologies as well as computer science and other related fields to transform the way that we think about these problems. So I'm going to skip a couple of slides and just actually tell you the end result. So we've been doing this type of things for a few years. And what we've recently found is that we were able to do these completely automated screens, meaning we just tell the computer to go, and here is a population of mutant worms, and the computer will make a decision on which worm is interested in saving or which worm is not. You can imagine doing this exactly the same thing with drug screen, so essentially giving you scores of how the drugs rescue a particular phenotype. So we were able to find several mutants that are actually very similar to what the biologists that we're collaborating with were able to find in his lab in the last 10 years manual research. So we could do this type of screens in a couple of months as opposed to a couple of decades to conduct these large-scale screens. And the most interesting thing to me is actually we were able to find certain mutant phenotypes that by eye they had never seen in the last 10 years or so by manual screening. So this tells you the advantage of doing things in a quantitative way and the advantage of being able to use technology to your advantage, to do things that are not subjective. Okay, so I will mention one other well, this is a microtechnology, but it's not really quite a microfluidics application that we had in my lab, which is actually to use this tool called Optogenetics. How many people here are familiar with Optogenetics? Okay, so maybe I'll explain a little bit. So Optogenetics is a tool that's been developed essentially to genetically engineer photon-sensitive ion gates or ion pumps into muscles or neurons. And by doing that, you are able to transform these cells into cells that can be controlled or gated by photons. So you can use certain light to turn on the cells, and you can use certain other photons to turn off the cells. And this is a way to directly interrogate the nervous system and potentially the muscle system. So the tricky part with C. elegans using optogenetics is that the worms are so small. They're only about 1 millimeter long. And in a lot of situations, people do optogenetics by taking a fiber and then chasing after the worm under the microscope and shine the light on the entire worm. So there's no specificity of which cell that you can activate. So that type of experience is not entirely informative. And what one of my students actually did was to actually incorporate this already available microtechnology, which is from a projector, and integrate that with a microscope system and allow you to directly control the activity of neurons. And that's pretty cool. So I'm going to show you one silly movie. This is essentially just to illustrate that we could do it. So the worm is sitting here. It's soaked in the drug, which actually immobilizes its motor neuron activity. So it's not going to move voluntarily. But then we can actually shine light on the muscles directly and control the contraction of the muscles. So this is a completely automated and feedback system. And basically, we're just telling the worm to bob its head. And we do that with the worm's tail. Now the worm's wagging its tail. And then we can make the worm sort of move in a pseudo-naturalistic way as they crawl on our plates. So you can make these shapes. So this is a direct way to interrogate the muscular system. That's not really very, very useful unless you're screening for muscle drugs for some reason. But one example that I would give is actually we use this system to look at a circuit that controls locomotion in worms. So here's an example where we shine blue light to turn on the neurons that normally tells the worm that it's been touched in the head. So we're touching the worm with a blue light. Now the worm goes backwards. Now we're going to also turn on, or we're going to shine green light to actually turn off the downstream neurons. So you will see that the worm starts to go back, and then you turn on the green light, it puts a brake on that behavior. And you can actually do this in multiple cycles. So this is now becoming a very exciting tool where we can actually interrogate the nervous system, how circuits behave, how behaviors are produced, and you could use this to test all sorts of hypotheses in terms of how genes and environment and drugs can actually produce behavior or control or modulate behavior. Okay, well, I'm not going to belabor this point, but this is a technology that we just directly borrowed from what is on the market and integrated it with our system. So I'm going to show you one more example on optogenetics. This is actually a potentially drug screen type of platform. So here we have a microfluidic system where we can load multiple worms, and then we can turn on. So watch when the thing says blue light on. You see what the worm did when the blue light turned on? So it all actually kind of shrink. So this is a particular screen where we actually put the optogenetic reagents in the upstream motor neurons in the worms and then measure muscle output, muscle contraction. So if the worm actually has a neuromuscular disruption, if the muscles are not talking to, the neurons are not talking to the muscles properly, then you will get abnormalities in this shrinkage behavior. So this is a quantitative assay where we actually use microfluidics and optogenetics to be able to interrogate synaptic transmission, so how the neuromuscular junction actually behaves. So you can use this to do a lot of genetic screenings and potentially drug screenings. Right, so the last thing I think would be of interest that I'll show very briefly is that we came up with the technology to image cells as well as embryos. So here's an example of Drosophila embryo. Actually, yeah, so that slide that I skipped earlier, I don't know why it jumped, is actually a problem where we're interested in dorsal ventral patterning of embryos, for example. So we want to image lots of embryos simultaneously, and how do we load them, and how do we do things to them? So here's a device where we can load hundreds of embryos in suspension into these chips. So the embryos are in suspension, and they get pushed into the device, and then they lodge into one of these traps, and eventually by simple fluidics they get flipped into the vertical position, and then we can image a lot of these things. So using that system, this system loads very fast. It loads in about 2 minutes or so, so you get a whole chip full of embryos that are correctly positioned. And I'll show you some of the results. So we can actually go in and look at the morphogen gradients and do these quantitations of where they are and how they interact with each other, et cetera, et cetera. So this is a really good platform for us now to look at multicellular systems, including embryonic bodies and embryos of different species, and see how these morphogens signal transduction actually happens during development. And you can imagine using this to screen drugs as well. Okay, so last thing I will show you is a cell trap. This is a device where we can actually load, these are T cells, and you can actually The movie is plain, probably a little hard to see, but again, these cells are actually fallen into they're in suspension, and then they get pushed through the device, and they actually dock onto these little trapping locations. So here's a fluorescence version to show you where the cells are. Now they come in and they actually deterministically dock onto the traps, what we call traps. So these cells can be arrayed in a matter of a few minutes, and we can actually have 2,000 of these cells in the chip that is on, well, the total dimension of that chip is probably about 1 by 4 millimeters or so. It's a tiny little area. And then you could go in there, you could do perturbations to the cells, you can stain the cells and then watch either activities of the cells, or you can actually fix the cells and stain them for something of interest. And the other beautiful thing is that because this is microfluidics, you can integrate it with other modules upstream to stimulate the cells or to do something to the cells in situ. So I'll show you one example of that, and then I'll be done. So here's an experiment that I did with Melissa Kemp's lab in looking at the calcium dynamics in T cells. So these cells are stimulated by hydrogen peroxide, and you can see that even from the same isogenic population, these cells would actually respond very differently. And you can quantify how the cells behave, and this would be very, very useful in looking at how patients respond to a particular drug, or how heterogeneous population from particular patients are, and these have implications in drug-resistant cancer, and also in behaviors of the immune system in general. So we're also taking this approach to many other applications that involve cells and imaging cells. Okay, so I am running out of my time, and I just want to say that here at Georgia Tech, I think we have a lot of opportunities to put these technologies together with the right applications and really thinking hard about what technologies might come together to solve the relevant problems. And I think with that, I will end my talk and see if there are any questions, and then we can get going with the rest of the interesting discussions.